Recording. I'll screen share. Are we good? Any issues? Okay, guys. And your questions there last week, just self-reflecting and all that. And yeah, the last week and this week are kind of biologically, can't say unrelated, but yeah, you know what I mean? We all want to get to the organ systems. We all want to get to all this kind of stuff, but we can't get to the bones, the heart, the kidneys, all this until we get this stuff done. So that's why I've made a test. This test for next week is only on the first 2 weeks. It's kind of pre-organ system stuff. So here we are. Let's go. This is chapter 3. And let's blow this up a bit and move this down. These are just all the things there that we're going to be talking about. Labeling parts of a cell. Like we have all of our different organ systems in our own body, but a cell functions on its own. It's independent. It can't exist on its own, but it's got all these different organs per se. We call them organelles, and being able to label them, being able to understand their function and all this kind of stuff, we'll get in there. We're also going to talk about biology as a brand new language, so we're going to go into some words. We're going to talk about mitosis. Okay, this is cell division. We're going to get into cell differentiation. You guys ever heard of stem cells? It's the new science. And but again, where we're at is if you take a flat stone and throw it against a still lake, it's just skimming the surface. That's all we're doing. All right. And great words here. Apoptosis. We're going to get into all these and all these kinds of things. So really good, really important chapter. And I'm going to move on. If I'm going too fast, slow me down. If I'm going too slow, say in, hurry up. Cells are specialized. And you can see here, like here's an RBC, red blood cell. Okay. These guys here, they only live for about, I think it's 3 months. And it's one of the reasons why our feces is brown, because most of it is RBCs. Okay. There's no nucleus. And all these kinds of things. We'll get into these when we get more into blood. But you'll see here, you'll remember there from last week your definition of anatomy and physiology, right? Function and structure, these kinds of things, they are tied together. And if you look at a nerve cell right there, there ain't nothing else that looks like a nerve cell. And if you look at its structure, completely tied to its function. What does it do? Send messages. It's a railroad track. It moves, and there you see it right there. So, this is a typical cell, okay? And you'll see, you know what I mean? You really don't have to memorize these things, again, for a testing situation. It's good to have them in here, all right? But I'm more interested in being able to recall and find it. And so all these different things here, absolutely responsible for. You'll just see here, I'll just kind of move things around haphazardly here. But here's your nucleus. This is the brain, right? And this is where the DNA is. DNA, deoxyribonucleic acid. This is our genetic blueprint. That's what makes you, you. And you. And me. All right? And we'll get into all that there today. But that's your nucleus. You'll see there, there's a nucleolus. See, I'm throwing words out here, and hopefully there 30 to 40% of them will stick against the wall. And then you guys can go back there later on in the week and review this again and try the practice questions, and you know what I mean? Then you guys will be up to 80 or 90 or 100%. Okay, so that's what we want. So you've got the nucleus. There's a nuclear membrane. Here you'll see it's very porous. And if a membrane is porous, that means things are moving in and out. Okay? You have your mitochondria. These are your powerhouses. These are your batteries. This is where the energy is made. You've got things called centrioles, microtubules. And you look at these ones here. Oh, my family's leaving and my beagle's going nuts. I want to go. So, yeah, it's all right. You look at these microtubules. These are for scaffolding. This what keeps the cell this shape, because a cell is just a membrane, which is just like a piece of cellophane, plastic wrap per se, you know, it's very thin, there's nothing there. And what gives a cell its shape? Well, you'll see here you've got these microtubules, okay? And there's a cell membrane. There's ribosomes, which are involved in protein synthesis. The fluid matrix, okay? You know, you think like a cell is like a jellyfish. Jellyfish, if you take a jellyfish out of water, you know, this huge man-of-war jellyfish, if you take it out of water, you could hold it in your hand because this thing is just so big because the water envelops it, right? Well, this is what's going on here. This is called your cytoplasm. I may have to go and take care of my beagle and throw it outside. You've got lysosomes, okay? You've got Golgi Apparatus. You've got Endoplasmic Reticulum, which we'll say ER, okay? You've got rough and smooth. There's all these different things here. Again, don't panic. It's not about memorization. It's about being able to find it and get through it, okay? So when you guys get your textbooks and all these, you can see these, but this is what we're looking at right now, okay? Oh, you poor dog. Oh, I know the whole family left. Oh, well, come on in, you poor dog. I know. Well, I'm teaching a class, so just sit down and relax. Don't puke like last week. There we go. All right. Here's just another shot. I think this one here came from your textbook. Okay. And this is just another shot diagram of the same stuff. There might be some new things in here. Don't worry about any of that. And remember, you have to think the amount of information that I'm asking you to know, and then I'm going to give you a 50, 60 question test. So you have to think, what's the most important things that I'm going for? And then you want to look towards the questions there that I've provided you. Diagrams are huge, these kinds of things, okay? So let's move on, shall we? Why is that going like that? There we go. It's a little better. And that's a little high. Okay, good enough. So the cell membrane, it's what we call a phospholipid bilayer. Lipid means fat, okay? And we are 60 to 70% water. And water and oil do not mix, all right? If you take vegetable oil and water, the vegetable oil will completely separate from the water, and it's almost to the point where it's afraid, get me away from this water. And the water is like, oh, get me away from this stuff. Okay? So you've got a fatty membrane, and you'll see here, this is the membrane itself. All right? And this could be the, it says right here, extracellular space. So this is outside the cell. This is inside the cell, if you can kind of get my frame of reference here. Okay? So there's going to be water in here in the cytoplasm, and there's going to be water outside, right? So you look at these here. If you zoom into your membrane, all right, and you see here I have my water liking, hydrophilic. These are all, they're cool with water. This side here of this is good with water. And you'll see here these fatty lipid chains, okay? These guys here are afraid of water. They're hydrophobic. And this is how membranes operate. Okay, so because some molecules don't like water, some molecules do like water, and how they move through membranes, because that's how the entire body works. It moves from inside to outside, and then it gets taken somewhere else, and then it goes from outside into inside. So things are moving across membranes. You know what I mean? That's the way things work here. So you have it's called a phospholipid bilayer. It's 2 layers, right? Okay, that's how this thing works. And you'll see here that there are protein embedded actually within the membrane. You'll see all these things here. And when we get into virus, and I think that's what when we do blood there, when we do immunity, you'll see here that there's a lot more going on in the membrane than just a piece of cellophane, a piece of plastic wrap enveloping something. Okay? There are these proteins embedded in it that allow things to come in. And not everything can go through this particular protein. Maybe this protein, it's like a bridge between the inside and outside. It only lets certain things in. And then this one here will bring certain things out. So it all depends. And you'll see here these are sugar, little sugar molecules here. And what they'll do is white blood cells will come around. I call these the sniffer dogs, the killer T cells. T cells. These are the white blood cells there that I call them the sniffer dogs, because basically, as the blood is everything is moving around, these guys here, excuse me, they're sniffing all the cells. They're reading every single cell as it's moving along to make sure that it's one of ours. Because if it ain't, all of a sudden now the alarm bells go off, and this cell that is a foreign body, okay, it gets intact and engulfed, if at all possible. All right. So, and how do the sniffer dogs, the T cells, find these? It's these guys right here. But we'll get into all this there later. Okay. So, phospholipid bilayer, that's what we're looking at. Protein molecules, they're channels to move things in and out. Semi-permeable. I like this kind of stuff here. Inside the cell, we'll start off with the nucleus. Okay. There's the nucleus, which is the brain per se, but then everything else we call the cytoplasm, and it's just the matrix of everything that's going on around in the cell. So here's your nucleus. Everything else in here we call the cytoplasm. Everything else, okay? So the nuclear membrane is porous. Things can move out. Things found inside the nucleus, there's the nucleoplasm. It's a gel-like substance. The nucleolus, which is involved in synthesis of ribosomes, which we'll get into. And chromatin. Okay, here we get into your DNA and your genes and all that kind of stuff. Okay. The cytoplasm, a gel-like substance found inside the cell, but outside of the nucleus. Right? Remember, there's nucleoplasm inside the nucleus. It's composed of cytosol and organelles. These are these little organs. Number 1, mitochondria. These are I've never heard it called a slipper-shaped. I'd more call it a bean-shaped, but I don't know what kind of slippers these guys are. But anyway, as we said there before, these are the power plants of the cell. Okay? And these guys here, they provide the energy there for the cell. You'll notice here that there are cristae. See if I can blow in and blow up on these right here. Maybe too much. And you see what we're doing? You're dealing here. There's the outer membrane. These are your enzymes here. This is your cristae. Okay? And they contain these enzymes associated with ATP production. ATP, Okay, adenosine triphosphate, you just write ATP. Okay, these are those energy molecules that once they're activated, bang, they're batteries. And once they're all the energy is used up in an ATP molecule, it now becomes ADP. And now it needs to be recharged and plugged back in the wall. And once it's charged, it'll turn back into ATP, and we can use it for work. Why do you think the number of mitochondria vary among cells? The number of mitochondria per cell varies because of metabolic activity. And today, I think it says at chapter 4 there, we're going to get into metabolism and energy production and what cells need. They have. You wait and see. It's a little bit of a long lecture, but it's actually crazy important. So a mitochondrium there, they have two membranes, okay, and that's what we're looking there. If you remember there, cristae, you remember mitochondria, power plants of the cell, and ATP. I think you'd be pretty good there. Ribosomes, these are making protein. And protein, the one atom that is involved in protein, muscle, is nitrogen. Okay, and this is, where did that go now? There you are. All right, so that's why you really can't get nitrogen from, you can get it from beans and these kinds of things, but it's not an easy thing to process. Like in a situation of fasting and you know, starvation, extreme fasting, the first thing your body is going to use is sugar. Sugars are the first thing your body can just soak those up real quick. The next thing, once all the sugar's gone, your body will go after fat tissue, lipid. Okay, huge calorie content, but not as easy to sugar to break down. Proteins, they are the most difficult to break down and the difficult to make, and they actually have their own process, which we're going to go into. Your body synthesizing proteins there. And ribosomes, okay, this is the site for protein synthesis. All right, and these guys here, we'll show you the picture of them there. Endoplasmic reticulum. This is a network of membranes. Okay, there's the rough ER and the smooth ER. Okay, they're different. So there's two endoplasmic reticulums. One is rough, one is smooth. I'll show you pictures there. The rough ER, there are ribosomes on the surface. Smooth ER, there is no ribosomes. Okay, so ribosomes are the place for protein synthesis. They can be found free in the cytoplasm, or they can be found on the rough ER. So the rough ER is for protein synthesis. There is also a smooth ER, no protein synthesis, no ribosomes, and this is a site for lipid and steroid synthesis. Okay, so ribosomes are protein makers. They can be found free in the cytoplasm or in the rough ER. Smooth ER is not for protein synthesis, it's for fats. Okay. What else we got here? So here's another organelle. It's called the Golgi, or Golgi, however you want to pronounce it, apparatus. And this is a package maker. Sometimes molecules, like you have to think, we're 70% water. Some molecules don't like water. So how do we move this molecule from here to my big toe? Well, let's throw an envelope around it. Okay, and you see here now, you see, you can make a package, and now this, whatever it is you're looking to get out of the cell, can now migrate towards the cell, and you see there that these things can now get out. All right, which they couldn't before because of an accessibility issue. So that's what we're looking for there. There's your Golgi apparatus. Okay, and you guys can refer all back to these two slides here. Everything is here. Like there is your rough ER and your smooth ER. It's all right here. Okay. Don't forget about cilia. Some cells there, they'll have a tail. Some bacteria, spermatozoa, these kinds of things. But we'll get there. Yeah. Is that? Interesting. Sorry. Sort of beep. The Golgi Apparatus is a series of flat membranal disc sacs. Depending on the protein's final destination, the Golgi Apparatus finishes the protein by adding or removing sugars and enveloping it in a package so it could move. Lysosomes, and you can see here, like this is all the information on lysosomes I think I'm going to give you. So this is other than the diagram itself. So these ones here, there might be a question. You know, what is a lysosome or something like this. So lysosomes there, they break down waste for housekeeping. And you'll see there that the body is crazy efficient, but nothing's 100% perfect. So you've got these guys here, and what they do is if there's any bits of junk or garbage floating around in the cytoplasm inside the cell, what happens is chomp, chomp. All right? Like Pac-Man goes and cleans up all the debris and cleans everything up. Okay? You have your, remember I was talking about the microtubules? It's almost like the scaffolding of the cell. It's the support structure for the cell, and we call this there the cytoplasm, or sorry, the cytoskeleton. Okay, it's composed of these microfilaments, microtubules, and shape and support. There is a centriole. These are rod-shaped structures. They help separate chromosomes during mitosis, which we'll get there. Okay. And look, we're already halfway through this one. So these ones aren't too bad. Microvilli. These ones here will be, you'll see them again when we do the intestine and the digestive tract. And you have to think, the your small intestine there, you know, parts of it there that are used for absorbing food, you know what I mean? They're not very long, what, a meter or two? And so how does all your food get digested out of a meter or two of intestine this big? How is your body able to absorb everything that it needs out of this? Well, it's because of these microvilli. And if you take a piece of the small intestine and just look at it, you know, here's your tube, right? And we've just taken a snap. And so you've got this tube here. And this piece of intestine, well, I'm holding it right here, and it looks like it's probably about 8 centimeters long. Well, yes, but the inside where all of the absorption happens as your food travels down through your small intestine, it's probably about, if that is 8 centimeters long, there's probably about a meter of surface area on the inside. And you see, your body is very good at taking a short distance and increasing the surface area. Because on paper, it looks like the surface area is only about 8 centimeters. But really, when you're talking absorption, that 8 centimeters could be a meter long. I'll show you all this and when we get in there. But microvilli, they're these little hair-like projections. And they're involved in water and nutrient absorption there in the small intestine. Cilia, hair, okay. A great example of cilia there is in the lungs, okay. If you have some type of infection or bacterial infection or smoking or whatever, what will happen is this is your broom, and these hairs are pushing junk, mucus, because your lungs have mucus, right? It collects all the dirt and dust in the mucus and then kicks the mucus out. So that's what these cilia are. Flagella, these are the tails. Best example is sperm. Okay. So that's all the organelles there that I'm asking for. And I think there's maybe 10 or 12 of them there. So they're there. They're contained in 10 slides. They're there. You guys will be good with those. Okay. Transportation, moving things around. All right. Your body is amazing at using free energy. And you have to think this is millions of years of evolution going on right here. And if you see, there is something called passive and active transport. So passive transport is using free energy. And just the whole idea of concentration, like if I take a little bottle of perfume and I go to the other side of the house over here, like I'm pointing like I have an enormous house, but really I'm just pointing over there because it's not my house. And I take a little and I spray a little bit of perfume over in the corner over there. In about 5 minutes, the whole room will be enveloped with perfume smell. And you see, that perfume moved from a very concentrated gradient, because I sprayed it right in the corner, and then it moved to a lower gradient, right? Things move from a high to a low, always. It's free money. It really is. And your body uses this passive transport all the time. Okay? You have a membrane. Okay? You have things on this side, you have things on this side. Well, what if I put a whole bunch of salt on this side of the membrane? Well, this side doesn't have any salt, sodium chloride. What's going to happen? Things are going to move from a high to a low, and things are going to become dispersed if let. Okay? The opposite of free money, of free transport, is active transport. This one here costs money to do, but it happens all the time. Sometimes we need something done. It's got to be done. So it's working against a concentration gradient. It costs money. It's not natural. It's not normal per se. So therefore now we have to inject energy into it to get this thing to go. Okay? Passive is free money. Active costs money. Okay? Types of passive mechanisms here. We've got passive diffusion. Okay? So diffusion, facilitated diffusion, osmosis, tonicity, filtration. These are all passive. Those are all free. Tonicity. Thank you. Whew. Haven't had enough coffee yet. Active transport. Okay, we've got these transport pumps. You've got something called endocytosis and exocytosis. Endo means in. Exo means out, moving out. So these are what we're looking for here. Please take note of an isotonic solution, a hypotonic solution, and a hypertonic solution. Okay, we'll get into those there. So here we've got diffusion, movement of a substance from an area of high concentration to an area of low. And eventually there will be an equilibrium. Okay, that's diffusion. Okay, your lungs, when we get into the lungs, it's just incredible how the oxygen content in the air is about 21%, which is high compared to the oxygen in your blood. So every time you breathe, the oxygen in the air just jumps onto your lungs. It's free money. And the carbon dioxide in your blood is higher than the carbon dioxide concentration in the air. So when you breathe, that carbon dioxide just jumps from a high concentration in the blood to a low concentration in the air, and that's how we get rid of our CO2. Works the opposite there for oxygen. We'll get there. We'll get there. There's lots to do. Facilitated diffusion here. There's a helper molecule, okay, that assists with the movement of the substance from a higher to a low. You remember those protein molecules there embedded in the membrane? There you go. It's called facilitated diffusion. Okay, so it just means that there's a helper molecule there to help. Osmosis. Just the movement of water, not particles. Here and here and here, we were talking about the movement of particles. Osmosis is not about moving of particles. It's about movement of water. So movement of water from an area with more water to an area with less water. So you have a look here. Here I have a membrane, and here I have a solution. It doesn't look that concentrated. There's some in there, but, you know, and then you got this side here. You got a lot more particles in the same amount of water than you do on this side. So this side here is more concentrated. So there we can say that this side here, okay, has less water, right? Per, you know what I mean? And so what will happen here, there's a higher concentration of water over here. So what happens is only the water moves. And you'll see right here, isn't that? And you'll see there that the concentration on this side is now the same as the concentration on this side. No particles, ions moved, only the water. Cool, eh?
May I ask a quick question in regards to the facilitated diffusion?
Yeah. Who am I speaking to?
Blake.
This is John, right? All right, go ahead.
I go by Blake, but yeah.
All right, Blake.
So I'm not sure because it says a helper molecule within the membrane. So
these guys, right?
The molecule comes from the outside, and if that's the case, do the things in which slide was it? Number 6, the cell membrane. The thing that scans all the cells that come in, does that not target it as an outside body and stop it, or
no? The when we get into the immune system and all that, the killer T's, that's a different one there. What we're just doing here is we're just talking about movement. That you need this information to get to where you're going, and we'll cover that there probably. I think it's week 5, but the facilitated transport here is you'll see these transport molecules that are embedded, these protein molecules that are embedded within the cell membrane. And what these guys here, they have a cap on the they have a cap on the top and a cap on the bottom, right? If it's closed like this, nothing's moving. But what'll happen is if there's too high of a concentration of something out here, what'll happen is this guy will open and these guys will come in and we'll start moving things through so that there's more of an even distribution. So that's what we're talking about.
That's what facilitates.
And it's free money.
Okay.
That's what. Hey, great question. I love it. So, I know we're right here. Yes. So this one here, the facilitated diffusion, again, it's all free money, okay? And in this case here, the things that you want to transport can't go through the membrane, so they go through these carrier protein molecules that are embedded. It's still free money, though, for moving them across. There is your osmosis. Osmosis, please remember, is just water moving, not the ions, not the particles, okay? And so that's what's going on there. Here we've got the ability of a solution to affect the volume and pressure within a cell. Now, have a look at these. These are always money on a test. Oh, did I just say that? Oh, cat's out of the bag. So here we go. You've got isotonic, hypotonic, and hypertonic. When a red blood cell is placed in an isotonic solution, no net movement of water occurs. So you see the amount of ions inside and outside the cell are about the same. So everything is good. So in a hypotonic solution, the water will diffuse into the cell. For a hypotonic, now water is going to enter the cell. All right, because there's not enough water in the cell. And so what happens, water will go in, and this is where we can get cell lysis, or the cell will burst, it will break. Okay, so hypotonic, you've got a very inflated cell, and most likely it will explode. Hypertonic, water will come out of the cell. All right, and these cells won't have any control over it. You know, like when I think there was a carrot in the back of my fridge there, and it was pretty bendy. It wasn't moldy or anything. It was pretty bendy. I just put it into a glass of water for a day, and then all of a sudden there the next day it was nice and crispy again, because more water outside the cells, not enough water inside the carrot cells. So what happened is water just came in. Okay? So that's what we're talking about there. Know those three for sure. Be able to recall them and differentiate. That's all. Okay?
So just to make sure I am understanding that properly.
Yes.
So pretty much if there's not enough, it'll take from it.
You remember these? You remember all these free types of transport? They move from a high to a low.
Yes.
So if you look at this situation right here, like this is the end result, that this cell burst. But if this cell, there wasn't a lot of water in it, and there are some cells that are, they don't have as much water in them as other cells, you put this cell into water, what'll happen is that water will just, because the water rushes from a high concentration outside the cell to a lower concentration inside the cell, and boom.
So in my terminology, it's hungry, so it wants more, so it's going to eat them.
Sure. And then you have the opposite here, where let's say we have a salt water solution. You put a cell into a salt water solution. So inside the cell is very fresh, clean water. Outside the cell is very salty water. What's going to happen? All the fresh water is going to go outside the cell, okay, and into there, and the cell is going to shrink. Things always move from a high concentration to a low. So it's
like it dehydrates.
Yeah, that's perfect.
Yep.
That's exactly what's going on. Okay, so that's a isotonic. Hypo means low, and hyper means high. Okay, so good questions. Keep them coming. Sorry, I'm just trying to keep this going on here. Filtration. Welcome to the kidneys. I'm not going to go too crazy on this right now. We will go crazy on it there. I think it's week 9. Movement of water and dissolved substances from an area of high pressure to an area of low pressure. So the kidneys, they command the heart. The heart does all the dirty work, but the kidneys, they filter the blood, and you need high pressure. I mean it. You need high pressure. And if the kidneys aren't getting it, they will get it. All right? And it's just a way of cleaning out the blood. And so that's what this is here. Filtration. Again, it's a free thing, but we need to have, it's free money, but we need to have high pressure in order to do that. And things will move from a high pressure to a low pressure. Okay. What do we got here now? Now we're into active. Okay, active transport. These all cost money. All right, and they're all over the place. Nerve cells, muscle cells. Remember I told you there last week that calcium was, you need calcium there for a lot more than bones and teeth. Well, there's a lot more involved, and we're going to get into that. So active transport mechanisms, they require an input of energy. They pump. As passive was from high to a low, active transport is from a low to a high. So we're going against what these things want to do. Okay, so all these ones here we just did, they all went from a high concentration to a low concentration. And it's free money. But when you get into active, you're going against that. And you're going from a low to a high. And the only way to move additional amounts of potassium into a cell is to pump potassium in. So this is what we kind of do. You're going to have a membrane with sodium along one side and potassium along the other. Nerve cells, muscle cells, they all work the same way. And if you don't have potassium here and sodium here, well, you can't get any traction. You can't get any movement. You can't send a message, right? So all these potassium on this outside of the cell and all the sodium on the inside of the cell, it's not natural. They don't like being there. They like to be mingling and schmoozing and being with you, but they can't, because the nerve cell will not propagate. The muscle cell will not propagate. So we've got to force these ions in these positions here. Okay, so this is what we're talking about here. Cost money, cost ATP. Endocytosis. Okay, this is the ingestion. Okay, let's say you have some type of foreign body here, and there are certain cells there that go chomp chomp. They eat things. They clean things up. They get rid of bad things, bugs, this and that. Well, what happens is here, whatever is to be enveloped, you know what I mean, gets the chomp. All right, and again, this is not natural. You know, this isn't like a ball rolling down a hill. This has to be done, and this costs money. Okay, and then right here we can do what we want with this. We've now created a vesicle here, and we can destroy it. We can utilize it. We can move it. We can do whatever we want. Okay. Exocytosis is the opposite. We're moving something out of the cell. It's the same process, just in reverse. Again, costs money. Cell division. Welcome to mitosis. But there is also cell division here. There's one called meiosis. Okay. Mitosis, we are dealing with body cells and repairing of body cells. Okay. Meiosis, we are only dealing with the production of sex cells. Okay. And this is the end result of mitosis is two identical daughter cells from the parent cell. Parent cell divides. In theory, the DNA is perfectly duplicated and separated. Two identical daughter cells are made, okay, from this parent cell. And this is how your body regenerates, arm, legs, these kinds of things. But meiosis, this is a little different process, and it all has to do with natural selection variability. And that's why you as a parent, if you can have 4 different children, and they will all look completely different, because we all have our own genetic predisposition. You know what I mean? Maybe I'm better at fighting off this disease, and you're better than fighting off that disease, and we call this genetic variability, and it's a protection against diseases out there. And that's why your children do not have all of your characteristics. All right? It's been split up, because if I give everything that I have to all of my children, then now if a disease comes and whacks me, it's going to whack all of my lineage, and in theory that's how species can go extinct. So, but we'll get into this. Mitosis is for body growth and repair. Meiosis is for sex cells. Okay? 2 phases of the cell cycle. You have interphase and mitosis. Okay? During the first gap phase, a cell carries on normal function and begins to make substances needed for cell division. There's something called the S phase, where the cell duplicates its chromosomes. Okay? And the final phase or second gap phase includes the synthesis of enzymes and proteins needed for mitosis. Now, we're not going into crazy detail here. You guys can see that there's 4 phases that are going on here. You have the G1, S phase, G2, and the M phase. Okay, and you see within the M phase here, there's telophase, anaphase, metaphase, and prophase. And we'll go into each of these. Again, we're not going into crazy detail. Okay, and you guys don't have your textbook, so everything you guys are getting is from the lecture, okay, from the PowerPoints there and the questions there. So don't go too crazy on this stuff, because remember, your test is going to be, 50 or 60 multiple choice. How deep can I really go on all of these, right? So, good. The M phase, which is this one right here, we're going to break this up, okay? And you'll see there that there are 4 phases. There's prophase, metaphase, anaphase, and telophase, okay? And you see mitosis, if you will remember, this is body cells, right? Here we're looking for an exact duplicate of the parent cell, okay? And you'll see here that the chromosomes line up and split into 2 identical cells, or 2 identical sets. And you just go on here. There's your cell, and it's starting to duplicate. All right? And now you've got double the DNA in here. So this is what happens here. You see, you've got 2 of everything inside the same cell. Everything inside doubles. And then things start to go to the poles, North Pole and South Pole. And the DNA lines up at the equator. And you see these spindles here, and they pull these guys over. And then you see here anaphase. What happens there is the cell starts to split. Okay. And here we go. Eventually, telophase there, we're almost at the end. And then you have your 2...
Are those known as daughter cells at this point, or is that something different?
No, these are daughter cells right here. So once you pass the prophase, metaphase, anaphase, and telophase, bang, you're into the 2 daughter cells, which are exact duplicates of the original cell there. Okay, very good. Good questions. Keep on. Cell differentiation. And you'll see here that these cells here are called stem cells. And ethically, some people, they have issues with it, all right? Because these are cells, that are associated there with, babies, fetal. And these are cells that can be turned into anything. Like, they are just gold. If you're talking biology and science and curing diseases and these kinds of things, what? I got a cell that I can change into anything? These are it. And you can see here that these stem cells, we are just learning the power of what is going on here, like muscular dystrophy and all these kinds of things there that, people become paralyzed. The biggest problem is you can't grow back nerve tissue. You just can't. And so once a nerve, you break a bone, that's the problem with breaking your back is, maybe you break some of your spinal bone, your spinal column there. But what happens is the spinal column will just like this. Well, you have your nerve, spinal nerve coming down, and this one, and it's just, you can't grow this stuff back. But the future is uncertain when it comes to these guys here. So these stem cells can go into neurons, nerve cells, RBCs, muscle tissue, bone cells, even skin, epithelial, these kinds of things. All right, so these guys are awesome. Those are stem cells. Relatively undifferentiated or unspecialized, whose only function is the production of additional unspecialized cells. Okay. The stem cells within the bone marrow and skin are capable of dividing more than once a day. Stem cells in adult cartilage may remain inactive for years. So they're the future. Ethical or not, I don't know. Disorders of cellular growth abnormal cell growth creates tumors. And just getting back into, let's go back into here. Now, this cell divides into two, and they are identical. That's what's supposed to happen. But when some type of foreign body, like a smoking, some smoke molecule from a cigarette, or gasoline, or some type of carcinogen, cancer-causing agent, comes in here, and it's in your blood, and it's usually a high energy, okay? And these high energy molecules, they'll come in, and what they'll do is at this particular point, what they'll do is they'll bump the DNA, they'll knock it, they'll damage it some way. So now this DNA, which is supposed to be completely doubled, it still separates, but now this DNA is not the same as this DNA. So now you have two completely different cells that are unrelated to the parent cell, and now these cells are rogue. Now this is cancer, because now these will start, now their DNA is not like it was supposed to be, it's different, and now they start dividing uncontrollably, making more mess, more cells, and that's cancer, right? So abnormal cell growth, these are tumors, these are cancer. Two good words there, benign and malignant. Benign is okay, it's not dangerous. Malignant is the bad one. Okay, that's the word you don't want to hear. All right. These cells extend and invade surrounding tissue. Here's another word I want you to know, apoptosis. Cell death. Okay, so benign, malignant, apoptosis. And that is that for this one here. Necrosis, death of cells or a group of cells. So apoptosis is the death of one cell. Necrosis, this is the tissue dying. Like, what was the name of that spider I was watching? A really good series on Netflix called 72 Dangerous Animals. And there was a spider there in, I can't remember the name. But anyway, you get bitten by... Sorry?
Probably Australia.
Yeah. Just trying to think of it. There was one of the spiders. It was just horrible, man. The spider bites you, and the venom just stays right there. And now, within a day or two, that little bite now, you got this much death. All the tissue around the bite is just falling off. And so that's what we call...
Could it actually be the huntsman you're thinking of?
Yeah, it might have been. The
huntsman spiders are harmless. They won't attack you. They get massive, but they won't attack you. They're just there.
I've seen those things. They're pretty nasty.
And they're massive, but at the end of the day, they're not there to attack you and harm you. They're just kind of like, hey, I'm hanging out.
I'm just
If
trying
anything, it's probably like a really small spider that you wouldn't expect, right?
Well, that's the smaller ones are the bad ones, right? That's what I've heard. But so this is what yes, the brown recluse. That's what I was thinking of. Yeah, it won't kill you, but it you can just Google brown recluse spiders, and you'll just get some. Just don't eat lunch while you're searching on Google Images for it. Yeah, go ahead.
I was just curious because you were on the topic of the spiders and that. If you get stung by a jellyfish, what happens kind of to your cells? Is it kind of the same as necrosis, where they just break down and start failing, or? Oh,
I tell you, some of those, and they're actually using a lot of those man of war jellyfish. There's a couple spiders in South America, even snake venom. They're using this as, they're trying to see if they can use this stuff for medicine, because some of this stuff, man, and I mean is lethal within a minute. And you're dead. And what'll happen is it attacks the nervous system, stops your heart, stops breathing, So, you know what I mean? You're basically suffocating. You can't breathe. It's a neurotoxin. Neurotoxin. Neuro is nerve. So if something's going after the nerves control everything. I can move my arms because this is a part of the, I can control these muscles. It's part of the musculoskeletal system, which we'll get into there next week. There's a whole lot going behind the scenes. Sorry I say that, Samantha. I'll get that in a second. There's a whole lot going on behind the scenes that you don't have control over. You eat that banana. You're not controlling your stomach. And you know what I mean? You're not controlling your ileum, jejunum, your small intestine. You're not controlling any of that. You can control your breathing, but only for so long. You know what I mean? My daughter there, like all kids, I think I did it as a kid. I'm going to hold my breath until you let me have that ice cream. All right, hold your breath. I'm watching you. Just make sure you sit on the sofa. Hold your breath. Okay. Pass out for 5 seconds. There you go. And you'll start breathing again. So there's a lot going on here. And these toxins, what they'll do is they'll attack the nerves. And if you're attacked the nerves, man, you're done. So there's lots. We're just on the tip of the iceberg. Ladies and gentlemen, what have we got here? It is 1 o'clock. Can you give me 5 minutes and I'll come back and I'll stop recording this and we'll move on to the next unit there, unit 4. So if that works, give me, let's come back at 1:10. Because right now it's 1:04, and go and get a refill, and we'll see y'all back in 5 or 6 minutes, okay? Bye, guys. Okay, so what I'll do, cancel, unmute, and stop video, and